Dr. McLean for tonight's introduction of Chris, and I will see you guys after the talk for the Q&A session. Have a good time.
Thank you so much, Mert. Well, good evening, everybody. I hope I find you and your family and friends safe and in good health this evening. I'm Craig McLean, the Executive Director of the Louisiana University's Marine Consortium, which is why we say LUMCON instead. And I'd like to thank you for joining us tonight for the 7th talk in LUMCON's online science series. Now, every Thursday night here at 7 p.m. Central, we're inviting a Louisiana scientist to share their research, and we'll give you the opportunity to listen and to ask questions of some truly amazing people doing some truly extraordinary science. I hope you will continue to join us each and every week as we explore more of the world from the comfort of our homes. And you can find more about the complete series, including our upcoming speakers, at our website, lumcon.edu. Just scroll down on our homepage to the link under News and Events. Well, tonight it's my privilege to introduce Dr. Christopher Green. Dr. Green received his bachelor's degree from the University of Oklahoma in zoology with a minor in history of science. Completed his master's degree at the University of Arkansas at Pine Bluff. Chris, what is the mascot for UAPB?
It is the Fighting Lions. So it is a lion.
That's nice.
A lion. Yep.
He went on to get his PhD from Southern Illinois University at Carbondale in zoology, and then took a position as a professor with Louisiana State University's School of Renewable Natural Resources. There he teaches a variety of courses, including principles of aquaculture, ichthyology, and fish biology. And if that doesn't give you a clue into his research, Dr. Green's research involves fish physiology. Man, that's a mouthful, isn't it? With a focus on fish reproduction, environmental toxicology, and salinity tolerance. And we're really excited to have him here tonight with us to talk about why we should all care about toxicology. And Chris, I'll turn it over to you.
Thank you very much. Thank you very much. I'm really glad to be here and talking. I'm a real big fan of these kind of seminars, and I'm really excited to be talking to you guys about toxicology. I'm going to try to get you interested in toxicology, hence what is toxicology and why should I care, and roll that into the kind of things that we see with a toxicant exposure, like say oil, and what it might mean for something like reproduction. As Craig talked about, I did my master's at the University of Arkansas at Pine Bluff, which is an HBCU in Southern Arkansas. And I actually started my career in toxicology, or in environmental toxicology, at that institution, where I investigated the toxicity of a breakdown product of Sarin Gas as a research project in conjunction with the Army Research Office and a arsenal down there that was storing. It was actually the second largest storage site for sarin in the country, that has since been incinerated, thank goodness, because of the Chemical Weapons Treaty Act of 1997. I started to talk to the public, like high school, elementary students across coastal Louisiana a few years ago, because I wanted people to know about environmental toxicology and what it might mean and what that career might be. And so the first part of my talk is going to be kind of introducing you into toxicology. So when we think about science and we think about the stuff that, if you've seen a lot of these seminars with LUMCON throughout the weeks, we see different levels of organization. So when we think of scientists in terms of, ecology and biology as a big science, geology and the like, we think of different regions, from say the entire world to specific ecosystems, such as say the Chafalaya Basin, or within there, if we kind of think of, our neighborhood where we live and interact with our neighbors and get our mail and go, sleep and stuff, we think of our community. And then when we think of that in terms of, thinking ecologically, we think of a population. So those groups, in terms of, like, here we have those largemouth bass, that those fish are interacting and they're living, growing, and reproducing as a group in that community, in that neighborhood. Well, for me, as an environmental toxicology person and a reproduction person, I work at the organismal level and inside the organism. So here is really good, a couple examples of some organisms. We've got a human right here with a big thumbs up. And then we've got, one of my favorite hybrid animals, the zonkey, a zebra-donkey hybrid that I ran across a few years ago on the side of the road and was like, I have to take a picture of this amazing animal. I work inside the animal as a physiologist and toxicologist. And when we think of those levels of organization, we think of organ systems. So, one organ system you might think of is delivering oxygenated blood to our body. The heart, the lungs, or if you're an aquatic animal, your gills, and then like the capillaries and things that supply oxygen or remove carbon dioxide, and also used to transport a lot of chemicals through your body. Now, when we think of an organ system, we begin to then partition into organs. And you might have in maybe a biology class in the past, or just, in regular conversation at a party, somebody might say, did you know that fish have a 2-chambered heart? Reptiles have a 3-chambered heart, and then we have 4-chambered hearts. And this picture right here, I want to point out the atrium and ventricle of the fish heart, but also take a look, there's a couple of other chambers here ahead and behind those two things. So when you think, when somebody says, oh, those fish with those 2-chambered hearts, maybe not so just 2-chamber. There are some other chambers involved. When we think of what makes up that organ, the heart, we begin to think of tissues. And this is actually a really good microscope picture of a tissue, the muscle tissue of the ventricle of that heart. When we get in a little bit more resolution, and I'm working with a lot of cellular and tissue kind of stuff, and we'll talk a little bit about tissues later. Think about cells, and these are the striated involuntary muscles of the heart, and you can see these dots here. Those are the nucleus of the cell. And anybody that might remember, maybe if you had 7th grade biology in this state, you had to make a model of a cell, and this is, thinking about the organelles in that cell. So all the nucleus and the mitochondria and the Golgi apparatus and stuff that make up the operations of that cell. Now, there's a few other levels of organization that are even smaller. And this is out of my realm, but we need to think of molecules that make up these structures. These are the 4 molecules with these little cool smiley faces. These are the molecules that make up our DNA. And finally, on the molecular level and beyond, when you think of like chemistry class and the periodic table, we're working with atoms that actually make up these molecules and thus beginning the whole chain of organization. So think about for a second, when you think of the word toxicology, what do you think of? What comes to mind when you think toxicology? Well, one of the things that you might think of is certain toxic events or certain, environmental catastrophes. About a decade ago, unfortunately, the Macondo oil rig actually exploded. And there was a loss of life, and it was a very large, tragic ecological event for the Gulf of Mexico. And throughout a very protracted amount of time, that well released around 210 million gallons of crude oil at an extreme depth. That then traveled up into the water column and dispersed a large area of our coastal ecosystem. And even though the ocean and the Gulf of Mexico is really, really big in volume, we saw a lot of this oil come in contact with a whole lot of animals and people that live in this environment. So modern history has led to a number of really, beneficial chemicals and things for industrialization. And so we utilize chemicals very often in, our own lives. But when we think about toxicology, we really, you can point towards certain chemicals that were useful to humans, but also had detrimental side effects. Some of the early ones in our industrial period could include, say, DDT. DDT was a chemical that was developed right before World War II. and it was actually instrumental in removing things like malaria and yellow fever from areas that were, occupied in tropical regions during our armed conflict in World War II. Unfortunately, it had huge detrimental side effects to a number of different animals and would bioaccumulate up the food chain and produce some really detrimental effects. Thalidomide is actually an anti-nausea drug developed in the 50s. The FDA actually didn't approve it, but a number of other countries utilized it for nausea. And unfortunately, it led to severe developmental effects where women who were pregnant that took this drug for nausea, the developing embryos and later babies would actually have missing limbs and things, and it was actually removed from the market. Later on, during the environmental movement, during the foundation of the Environmental Protection Agency, Silent Spring by Rachel Carson, who actually highlighted the dangers of DDT. The Cuyahoga River actually caught fire about 3 or 4 times because of the amount of industrial effluents and pollution that were put into that. And nowadays, the Cuyahoga River is actually championed as a big restoration success in terms of a polluted site that attention has been paid to the point where, it doesn't light on fire and actually sustains a good number of fish and insects and other animals. And finally, a more recent toxicological event, which is actually really, really big and important for our region in terms of our economy and livelihood, is Deepwater Horizon about a decade ago. So toxicology comes from the Greek word toxikos, which in Greek is poisonous. So as an introduction, toxicology is really the study of potential detrimental effects of substances on biological systems. And there's a lot of other disciplines within toxicology. And keep in mind that, when we talk about an ecological system, a population, an organism, or the organ systems inside there are different avenues of toxicology that begins to address a lot of those different levels of organization. One of the earliest evidence of, an understanding of toxicology comes, around 3,000 to 4,000 years ago, where we begin to see humans utilizing toxic compounds with knowledge of their use. An early example of this is from Homer in 1850 BC or 850 BC. The Iliad makes numerous references to the use of poison arrows in combat. Now, you might think of Homer as this, philosopher, but also Homer Simpson also has, some interest in toxicology, as he spends a lot of time at Moe's Tavern, which is also an aspect of toxicology, if you know what I mean. Socrates was actually sentenced to death for corrupting the young in 399. And he was actually, through that democracy, he was actually sentenced to death by drinking a potion of hemlock for his death sentence. And that was actually, in addition to warfare, we begin to see ancient societies understanding toxic properties in a real basic way. More formalization, or, kind of the modern, father of toxicology would be Paracelsus. He was around in the 15th and 16th century, and he really formalized the belief and kind of the knowledge that all substances are poisons. It just depends on the amount of that poison and the amount of what he called the dose to determine its toxicity. And so the biggest concept in toxicology, if you don't have any other take homes from this at all, and remember Homer Simpson at Moe's Tavern, and remember the key concept in toxicology is that all things are poisonous. Only the dose makes it non-poisonous. So the dose determines toxicity. So whether we're talking about synthetic or natural substances, all of them have the capacity to be toxic. You'll be like, well, what do you mean? Like, what's that mean? Well, toxicologists actually have been able to take this even further and create a dose-response relationship. And so in this diagram, the yellow arrow points to this part of the graph that toxicologists use as concentration. So across here, you have, say, increase in concentration of, say, salt in your body or some other compound. And then toxicologists often want to measure that concentration as, it increases in terms of a potential effect. And a lot of the effects that toxicologists kind of can see would be, the biggest one would be like percent mortality. So the amount of animals that might die from increasing salt concentration in their bodies, or say the amount of, some other substance in their food or their water. And we often see graphs like this with this line where the effect and the dose have an intimate relationship with each other. Now, you may be like, well, not everything's toxic. You're made up. Well, if you remember the Nintendo Wii, which I think has fallen out of favor, I haven't heard anybody talk about a Wii in a long time, but there was a contest back in 2007 called Hold Your Wii for a Wii, and a woman actually overdrank water in this competition, this radio contest to win a Nintendo Wii, and she actually died from water intoxication. And so when you think of substances that you're like, well, that's not toxic. Yes, indeed, even water, too much water, can be a toxic substance. Here's some other things that are everyday substances that you might not think of with being toxic, but they are because of that dose. Good example, I've picked out aspirin, vitamin A, and oxygen. So aspirin, if you have a headache, you'll be like, oh man, I gotta take an aspirin. Usually those beneficial doses are between 300 and 1,000 milligrams, very, very tiny amount, really tiny pill. However, we begin to see liver toxicity and other problems above 1,000 milligrams to 30,000 can actually be really detrimental. Similarly, vitamin A can actually produce toxic effects, can actually make you sick and hurt your body at higher concentrations. And finally, oxygen. Our oxygen that's surrounding us right now is around 20–22% oxygen and then mostly nitrogen. When you begin to increase that oxygen to 50–80%, it can actually damage the alveoli in your lungs. And so again, that dose, the concentration, can produce a potential toxicity or a potential benefit. Now, a lot of toxicologists deal with a few different concepts that I want to introduce to you in a broad way. One of the biggest one, past dose, is risk. Risk is defined as like the likelihood of an injury or disease resulting from an exposure to a potential hazard. And so all the things that toxicologists assess, whether it's a new drug or say a compound they might put in a food item, they look at the risk that might be involved in adding that substance to something else. There's evaluations of risk that always surround the basic concepts of toxicology. A lot of those risks can involve the dose, can involve the exposure time, or even how that potential compound might get in your body. Now, one of the first things I talk about when there's chemicals all around us, like even in this room I'm in right now, there's different exposure routes. And toxicologists often think about those routes as being, ways that those chemicals can get in your body. So some of the things that, you might, from your day-to-day just existing, you might have chemicals that are around you, say, outside or in your home or even in your school. Some of those places, like workplaces, if you're around certain chemicals, those are opportunities for you to be in closed spaces with chemicals that might, come in contact with your skin or might be in the air. Dietary, there's actually potential exposure from, say, compounds like pesticides and things that you would eat because of their application to certain food items. Accidental exposure, whether there's, say, a spill in an office place, or if you're working on your car and you get substances on your body, those are a route of exposure. And finally, like I talked about Homer Simpson, there are very good examples of deliberate toxic exposure. Say, when you go down to Moe's Tavern. First thing, when we think about where those exposure pathways are occurring, people think of, how a chemical can produce a biologic effect. It must to get into that animal. And so that's that exposure. Past that, toxicologists think of how that chemical gets in the body and then gets to certain target sites. So if we thought about that oxygen example, with too much oxygen, the damage from that oxygen would be, like your lungs, or say the aspirin. Once the aspirin gets into your body, it causes damage, say within your liver or your kidneys. A lot of that is under what toxicologists call toxicokinetics. And you can think of that similar to like what pharmacists do, pharmacology. There are a lot of toxicology and pharmacology programs in medical schools that whole semesters are dedicated towards how chemicals get into the body and reach target organs to produce their effects, whether it's your lungs or your heart or your brain. Also, we talked about the dose. Pericles was really smart about the dose makes the toxicity. So when we think of dose, that's obviously the quantity of that substance, but also there's the importance of time. So when you're exposed to a toxic compound, the dose is important, but also the amount of time that chemical is in your body at those target sites, doing whatever deleterious or bad things might be happening. And finally, body weight. So body weight plays a really big role in terms of the amount of chemical, how it gets in your body, where it travels to, and the size of your body overall. So think of that if you ever have to go to the vet and get, say, a flea medication for your cat or dog. You don't usually get the same big pill for a big dog as you get the pill for, say, a small cat or small dog. So basically thinking of the body size as a container, small versus large, often has a big role to play in terms of what that dose or amount has in the body rolling around. Finally, before we get into cool fish toxicology stuff, I want to talk about exposure and then the relative, amounts. What does it mean when scientists say certain amounts of compounds? Time of exposure is really important. So how long an organism is exposed to that chemical, whether that organism is exposed to a chemical that goes into an area and then disperses through that big, say the ocean, or if that chemical hangs around or is renewed with constant, say releases of that chemical from, say, the soil or other medium. Toxicologists often, I'm going to use a couple science words here, they think of duration and frequency and dose in terms of that exposure in an acute exposure or chronic exposure. So when people talk about an acute exposure, they think of like a single exposure, usually at a very high, very kind of pointed concentration, and then it goes away. And so acute toxicity is usually a demonstration of the most potent effects, the highest concentration you might see, when an animal, say, dying. A good example of an acute exposure would be if you're riding around in the marsh and you see a lot of dead fish from, say, a very, pointed low oxygen situation where, the fish have basically suffocated. That would be a real acute event, and then, oxygen would return to normal. A chronic exposure might be over a long period of time. So where you'll have a reduced oxygen concentration that won't be enough to, say, kill the fish. You won't see a fish kill, but that, lowered oxygen might have real, long lasting effects. So over time, we know that low oxygen, like say in a marsh situation, fish either move out of that area, or if they're exposed to low oxygen for a long period of time, it might alter their ability to, say, do reproduction and build hormones and things in their bodies. So I'm going to use a couple of units, some metric units, so don't roll your eyes because I've got some really cool examples of what that means. Toxicologists use these units per volume measurements like parts per million, parts per billion, and parts per trillion. And those are equivalent to milligrams per liter, and yes, that's the metric system, micrograms per liter, and nanograms per liter. And you'd be like, well, what does that mean? Well, you can think of part per million as one drop within, say, a bucket, just a standard Home Depot 5-gallon bucket. Parts per billion can be one drop from a little dropper in a whole swimming pool, which is pretty big. So when you hear of substances being extremely toxic, generally, the more toxic things are, the more effective they are at even higher, at lower concentrations. So something that's reported in parts per million is not as toxic as, say, something reported as parts per trillion. Here's a pretty fun few slides that I put together on what does it mean to be parts per million, parts per billion, and parts per trillion. Part per million, you can think of like 1 inch in like 16 miles, which is, that's a lot of miles. 1 penny in $10,000, and 1 bad apple in 2,000 barrels of apples. And then it gets even more ridiculous when you look at parts per billion. 1 cent per $10 million, 1 bad apple in 2 million barrels of apples. Okay, so very, very one little small unit in a whole lot of something. Finally, parts per trillion. That's 1 second within 320 centuries, or 1 bad apple in 2 billion barrels of apple. And also 1 grain of sugar in an Olympic-sized swimming pool. That's equivalent to 1 part per trillion. So when something is toxic or producing an effect at this concentration, it is very effective at doing its job. Now, this is actually a picture released from NOAA during the 2010 Deepwater Horizon event. And you can see on one side of the screen, there's a small oil slick along the Gulf of Mexico. One big cool thing is that the Gulf of Mexico is really big. So your oil, even 210 million gallons of it, gets, kind of dispersed or kind of distributes through that large volume. The difficulty is in the Gulf of Mexico, things move with currents. And so a lot of that oil is deposited across our real critical marsh habitat. So, when we think of 1 grain in a swimming pool, that's great. When we think of, oil moving towards the coastline, that accumulation can be bad. I also wanted to show this picture because you might think this plane is skywriting. Is it skywriting? No, it's not. It's releasing a chemical dispersant or a surfactant to break up that oil slick and get the hydrophobic, afraid of water oil broken up into smaller compounds, which aid in its digestion by microbes and kind of break down through natural processes as well as wave action and sunlight. For a second, I want to take a moment and introduce in an introductory kind of way, what's a surfactant? What is a surfactant? A surfactant is a chemical that helps oil and water mix. Now, you'd be like, surfactants are bad. Well, surfactants are very, very common in industrial chemical situations. There are thousands of surfactants on the market. Some of the ones like we think of with oil spills, like Corexit 9500. There are personal care products, so shampoo, conditioner. There are surfactants in food and medicine, as well as herbicides. All these things help get a hydrophobic or something that's afraid of water, some non-soluble compound, to get more soluble in a water or kind of water-like solvent solution. We first see these used among ancient peoples in the forms of animal fats and ash. So humans have been using surfactants to a certain degree for thousands of years. During the Industrial Revolution, we begin to see, organic chemistry utilizing in the 1800s, making surfactants. And then in the late 1900s, we begin to see people actually making microbes produce surfactants and harvesting those surfactants for commercial purposes. Some of the scary names you might hear of surfactants include sodium lauryl sulfate, diacetyl sodium sulfosuccinate, surfactant, FA glue, DOSS or diacetyl sodium sulfosuccinate. That's actually used in Corexit 9500, which breaks up oil spills. However, a related compound, sodium lauryl sulfate, SLS, is in your shampoo right now in your bathroom. So next time you take a shower, take a look at that shampoo bottle, and you will see these compounds are similarly named compounds because they're surfactants used in the cleaning of your own hair. You might use them every day if you take a shower or bath every day. So hopefully you've got kind of a background to be like, well, toxicology, that's some words. Now we're going to talk about fish, and we're going to talk about some research, and hopefully you're still interested in why toxicology. And we're going to talk about fish and fish reproduction and the role of oil exposure in that reproductive process. So I'm going to introduce to you the Gulf killifish. Back in our neck of the woods in Louisiana, along the coast, they're referred to as Kakaho minnow. For some reason, Mississippi, our neighbors, they call them mud minnows. And I'm not going to make fun of calling them mud minnows. I don't like the word mud minnow, but they can do whatever they want to do. They call them mud minnows if they want. Kakaho minnows is what you probably heard them as. I'm going to refer to these guys as Gulf killifish for our talk. They're an estuary top minnow. So these guys can live in really wide, low, and high salinities. So as it gets saltier or not salty, they can deal with it. They have a really long spawning time. They have a huge spawning period. They start spawning in early March. And as long as it doesn't get too hot, like above, say, 95, 92 degrees, they'll keep making eggs on a regular basis between every 14 and 28 days through the marshes in our coastal waters. As such, they create a really base food item. They're a really common angling item. So if you're an angler, if you go fishing, you might have put a Kakaho minnow on a hook to catch a redfish or a southern flounder or something cool. They're really, really abundant. So they're an important food web fish item. So they eat a lot of bugs, they eat a lot of small fishes, and they get eaten by a lot of stuff too. One of the reasons that I use Kakaho minnows or Gulf killifish is they can be kind of maintained and happy in captivity, so to speak. They have reliable reproduction. That means that I can hold them in temperature and sunlight and dark periods to make them spawn year round, and they respond really well to that. They don't get stressed out. They don't get sick as easy as other fish. They represent an animal that stays in the same place. And the nerdy way to say that is they have high site fidelity. I say they stay in one place. They're not going to migrate out. They're going to stay regional. They're such a tiny fish, you know. They're not going to be moving around. And also, a lot of previous studies use them in toxicology. Now, they're not like what you see in this upper part of this picture. They're not going to be out in the middle of, the middle of the Gulf of Mexico, where we get fish that reproduce, like this inset picture here. These are kind of the more smaller, delicate larval baby fish that you see out in the Gulf of Mexico. Those would be awesome to work with. However, I want to work with an animal that I know I can get reliable, healthy reproduction from. And also, here's a picture from South Pass. You can see the oil level, the oil line right in here with this with these grasses. They stay in that area. And as such, I think it's really important to look at animals that reside in the area that might get continual, possible oiling from, say, some kind of event like we've seen previously. So another thing is their range. Now, this top picture demonstrates their range across the coastal area of the Gulf of Mexico, and even on the other side of Florida and the Carolinas. They're not going to be in this open area, but again, they're going to be, as kind of the range of this oiling shows, they're going to be existing in a lot of these coastal habitats and a lot of these marshes and stuff along here. They're going to be around there, and they're going to have the opportunity, or kind of the fate, unfortunately, to get exposed on a regular basis. So one of the things is, how do you test the toxicity of oil? Well, we could look at the acute toxicity, that really high level of oil that might kill those animals. But one of the things in terms of, animals getting low concentrations of oil in their environment and, trying to live, grow, and reproduce, I wanted to see, what those animals might do if they have lower concentrations of oil. And so the concentrations of oil that I wanted to test are around the parts per billion concentration, or micrograms per liter. Unfortunately, oil doesn't like to get into water, and so it's a little difficult to assess. Also, oil isn't just one compound. It's a whole host of thousands of different compounds. So in the bottom of this slide here, what we have to see is that when we add crude oil to seawater, the ocean mixing, the wind, that energy that the ocean has, if you go get knocked down by a wave at the beach, that's energy. And what we see is the ability to produce a mixing environment to get what's called a water accommodated fraction. And that's the science nerdy way to say there's water, but there's very, very tiny microscopic droplets of oil that's in that soluble or kind of water loving component of that water, of that oil. So in nature, we get wave action and the tides and stuff that mix up that crude oil. But what we have to do in the lab is we have to mix that up in certain ways that replicate this environment. And so that's really the first step. And in my lab and other labs, we like to tinker with things. And so we like to build stuff. And I don't know if anybody here is interested in building things, but we found from previous research a really novel way to make a tank and then put a pump underneath that tank to circulate water through this system. And so what we have here is we have these containers and they have beads in them. And these tiny beads have taken up and soaked up oil. And then we run that water through there on a continual loop for say a day. And it takes the parts of oil that are more water loving than say water hating and mixes it in that water to create WAF, or water accommodated fraction. And this is our diagram of this guy right in here on the left. And then on the right, we have one that represents oil. So on the top, it's brown. You can see the oil slick. On the right of that picture, here's our control. And so we're demonstrating that we have water generated that just kind of circulates without oil. So we could create treatment oiled water. So we could create this water that we could use to expose fish to. And we could take, control water that was just salty water that we know our fish like to, hang out and do their thing in. So over a 40-day period, we took adult mom and dad Kakaho minnows and we put them in control and treated water. And we took 6 males and 10 females in 4 different tanks, clean tanks and kind of dirty tanks. And every 4 days, we took 50% of the water out of these tanks with these little cartoony fish in, and we put new oil in there. To be like, why don't you just put oil in there and leave? Well, we wanted that oil, the particles, the types of compounds in that oil to remain pretty similar over time. And so we had to replenish that, because if we just left it in there, it would degrade over time. So for 40 days, these guys had either oily water or clean water. Then after that period, we took those fish out and we transferred everybody into clean tanks that had no oil in them at all. So we're like, okay, guys, you had a lot of oil. None of you guys died. That's cool. Here you go. Have a nice clean environment. And in addition to that, we're going to introduce some spawning material. So out in the wild, these guys lay their eggs, the males and females, they kind of go around the spartina, the different grasses, and they lay eggs in the little nooks and crannies there. And so we have these fiber mats that we put in these tubes, and that's a really good home for the eggs. And so moms and dads, males and females, they go in there and they're like, hey, this looks like a cool place to reproduce. Let's lay some eggs. And so over a 40-day period, we put these guys in awesome water and we gave them really good conditions. And we said, hey, you guys spawn and make some babies. And they did. And they did on a really regular basis. We collected a lot of eggs from both oil exposed and non-oil exposed fishes. And each time we went into those tanks every few days and we took their eggs out and we counted those eggs and we looked at the larvae under microscopes to like measure them and stuff. And we could see the larval health based on their living or dying or their size when they hatched out. This picture on the bottom right here, the white line is like the length of the larvae, and we're able to see how much egg yolk, the little red circle here. And then we saw the depth of the little fish here. So we could tell a lot about the babies that the moms and dads were producing in these oil and non-oil exposed fish, which is really cool. Now, before we talk about those babies some more, how did the moms and dads fare in those oiled environment for 40 days? And nobody died. It was really low concentration, which was awesome. But we found that oil had some bad consequences on the dudes in that tank. And so here's our male Kakaho minnow, and he's like flexing. He's like, I'm a dude, I'm awesome. And on this graph here, I got on this side, the left, here's the oil exposed time. And we looked at their gonads. We looked at the male gonads at 0, 20, and 40 days of that oil exposure. And then we looked when they were in a clean environment in 10, 20, and 40 days after that, we looked at their gonads too. And we're like, how are you guys doing? Well, unfortunately, the dude fish, their gonad size decreased, while the fish that didn't have any oil exposures, their size of their gonads were the same the whole time. Then, good thing though, when we took those dudes out of the oiled water, their gonads increased in size, comparable to the non-oil exposed fish. So that was really awesome. But that might have some consequences for them. We also found that throughout this oil exposed period, at 20 days and 40 days, we actually found that those dude fish exposed to oil actually had reduced testosterone concentrations in their blood. And that's, if you ever listen to radio about, men having low testosterone, that might have some impacts on reproduction. Similarly, when we looked at the females, when we looked at ladyfish, we found that oil exposed fish during the oil exposure and even after didn't really increase in their estrogen concentrations in their blood. The females that didn't have any oil exposures throughout that period continued to increase the estrogen concentration in their blood. And we know by looking at fish, estrogen in fishes has a direct relationship to the amount of egg yolk or energy that female can invest in those eggs. And so this could have some really big consequences. So when we spawned these fish in that clean period, we were really excited because we found that the oil exposed fish had higher numbers of eggs. So those females were releasing more eggs. And we're like, wow, you guys were exposed to oil and you're putting out more eggs than fish that weren't exposed to oil. That's good for you. But there's a but in there. However, the females that were exposed to oil, although they had greater numbers of eggs, greater numbers and greater proportions of those eggs that were released, they died. And so we had less eggs hatched from females that had been exposed to oil. Now, I talked about estrogen, and we found that those females had low estrogen compared to non-oil exposed fish. And one of the things we saw is that we know that yolk, like this egg right in here, that yolk is deposited in those eggs as the amount of energy that that mom gives to that fish to survive, or in this place, a chicken. So the egg yolk is directly related to the amount of estrogen in that female's body. So the young the baby fish that hatched out, those who were hatched out from moms who were exposed to oil actually had less yolk in them to survive when they hatched out. And overall, they were smaller in size. So the size of the baby fish were reduced from from mothers who had been exposed to oil. And we were able to, this circle right here denotes kind of where we would find the yolk. And so when you think of yolk, like next time you have chicken or go to Waffle House or something, yolk is the energy that the mom can send to the baby for it to develop into a successful young, in this case, marsh-going kakaho minnow. That reduced yolk, unfortunately, made those fish have to eat sooner. So we found in our experiment that baby fish that came from moms who had been exposed to oil, they actually, when they hatched out, they were required to eat sooner or find food sooner, or they would die. Bummer. So one of my other take-home messages was Pericles, the dose that makes the toxicity. Well, here's some take-home messages in terms of this kind of oil and reproduction talk. The oil-treated adults produce more eggs, which we were like, whoa, that's awesome. But we had more of those embryos or eggs die, and we had less amounts of yolk or energy for them to survive, for them to develop into successful babies. The larvae from the oil-treated or water-accommodated fraction-treated adults, they didn't survive as long without food. So if you hatch out and you have to catch a small, like little bitty microorganism out in the marsh, if you came from an oil-exposed parent, you're going to be hungrier and maybe die sooner if you don't get something in your mouth as quick. We found reproductive impairment in terms of both estrogen and testosterone, and we saw that males had reduced gonad size as they were exposed to oil. But the good thing is they made up for that after they got out of that water. Also, oil exposures seem to be able to propagate in the next generation. And what I mean by that is when we had groups of parents that were exposed to oil, the consequences in the progeny or the babies from that were seen in the length of the babies, the amount of yolk in them, and the amount of individuals that actually survived. Finally, and I think this is really important, when you think of toxicity, when you think of toxicology, you might think of, fish kills or seeing dead fish in the marsh. But what I want you to kind of take home last is that toxicity doesn't necessarily mean that, you're going to see a fish kill. A toxic thing might produce small effects that, might not be immediately apparent with, say, maybe a few less babies, or maybe a baby that isn't as fit or healthy as it could be. So just think of that, because I think it has a lot of bigger picture consequences for your population, or for that kind of community or that kind of neighborhood that a fish like a kakaho minnow or others might reside in. The ultimate goal being to live, grow, and reproduce. So there's a lot of other experiments I could have talked about, but I really wanted to kind of break it down into this specific experiment and kind of what we saw from this. And I want you to understand that within science, it's not just like one person doing one thing and being like a one-man band. It takes a lot of different people to be successful and collaborate to figure out big picture stuff, as well as, simple things like what we saw in this talk. My lab, we conducted this research a while back, and it wasn't just me in the lab all hours of the day and night. There's a lot of other people involved, as well as some other labs and other people at LSU's campus and other locations. And also it takes a lot of funding from local, state, and federal support to get these things answered, to see what's going on out in our marsh and out in our ecosystem. So that concludes my talk on toxicology, why you should care, and role of oil and reproductive health. So I'd really like to say thank you to everybody for attending and giving me the chance to talk about this, because I really enjoy this topic. It's actually really fun, and I look forward to our conversation and anything that I can impart or talk about further if you have any other ideas or questions. Thank
and
you.
we do have some questions rolling in for you, Chris. First of all, I have to say, there were a lot of "this guy's great" coming through the.
Pardon? Sorry?
There was a lot of comments like "this guy's great" coming through the question box. So kudos.
Oh my gosh, I just forgot. I just forgot to give a Star Trek reference in my presentation. I was so consumed with talking about toxicology and oil, I forgot to talk about how Data or Counselor Troy would approach this or something. I failed. Oh my gosh.
That's all right. We'll forgive them, huh, guys?
No.
So we do have a few questions that have come in. And for the audience out there, we'll get to as many questions as we possibly can. So just be patient. If we don't get to your question, I do apologize. But there's a lot of questions coming in. So the first one was from Jonathan, and he's asking if toxicology can be used to predict the impacts of future disasters.
Absolutely. So there's a lot of what. So the group of toxicology that gets into that, there's a couple of different aspects. There is a group of toxicologists and modelers that can actually look at the molecular structure of a chemical or potential chemical and tell you its future effects on biological systems. So basically, I can show a toxicology nerd a molecule, and they can tell me oh, because of this configuration, I can tell you exactly what it might do in the body. It's actually really, really cool. It's not my thing, but that's a really good example of how toxicologists can predict, oh, well, if this gets out, where will it go in the body, or how will it react, how it will break down and stuff. Yes. And then there's also stuff where, based on research we saw with the cocktail minnows, if you sent me some water and it was, say, X amount of concentration of some kind of oil compound, I could be like, well, based on the dose response curve that I didn't show in this talk, I could tell you what that might do in terms of acute or chronic toxicity. Yeah.
Great, thank you. David is interested in whether bioaccumulation would be a dose or an exposure.
Dose or exposure. So the exposure would be any time. So bioaccumulation, for those that might not know, is where, and a good example is DDT. So I talked about DDT as a pesticide. It bioaccumulates in the fats of, say, bugs and other animals, and birds like osprey and eagles would eat it, and then it would impact the shell thickness of the eggs. And so an exposure, so the exposure would be every time that animal, say an eagle, ate a certain type of fish or insect with DDT, and then that would be an exposure event. The dose would be the burden or the amount of that DDT that's accumulated in that certain trophic level as it goes up. So basically, every time a fish gets eaten by a bird, that would be a dose and an exposure. You would have that animal is exposed to DDT, and it wouldn't get out of the body. It would unfortunately reside in the body, which is a whole aspect of pharmacology and toxicokinetics. And if you're interested in that, if you're interested in pharmacy school, that's what a lot of pharmacists and stuff have to deal with their first couple years of medical school.
Great, thank you. Tori's got a really interesting question about whether when a fish dies because of exposures to toxins, do the decomposers then have risk to being exposed to those toxins as well? Or is it so degraded at that point, it doesn't really have an impact?
That often depends that often depends on the type of compound. So for example, I talked about sarin gas. And sarin gas is a nerve agent that's highly potent within the receptors in the brain for acetylcholine esterase. The reason I mention this is it's very reactive. And so it produces a very acute event, but once it's in the environment, it breaks down and becomes a different substance. So the best way to answer that really good question is it's compound specific. So some compounds break down, they have a half-life. A really good example would be what we see in municipal water treatment plants with synthetic estrogens in birth control pills. So our estrogen and the estrogen in our bodies and testosterone, it breaks down very fast. Synthetic estrogen made by pharmaceutical companies is a little bit stronger and it doesn't break down. And so we see actual levels of synthetic estrogen downstream from, say, St. Louis or other big cities because it's very tough. And so, again, it would be specific to what type of compound you're dealing with. But yes, that potential could be there depending on compound.
Great. Got a fantastic question from Arturo. He's asking, do you think these results on cockahos can translate to other species such as game fish or redfish or speckled trout?
Yes, there are. They can. And I'm not the only one to do this stuff, by the way. I didn't wake up one day and think, I'm going to do this. I got the ideas to do this reading other people's papers. And there are people that have looked at stuff within redfish, mahi-mahi, speckled trout. You could get on something like Google Scholar and you could type in, like, say, spotted sea trout and reproduction and oil and find other examples of this in other species. I will tell you, killifish are very easy to work with in terms of larvae and stuff. Redfish, like red snapper and stuff, not as much. Hence why I use cockapoo minnows.
Aaron's got a question about how long the Wofe stays in the water column in natural settings.
So it's I'm not on the, so that's a good question. What I've read is a lot of it, so it does persist. However, a lot of it can get bound within the sediment. A lot of it can be broken down by microbes and stuff. And so, and dilution and wave action and stuff. So obviously, say if you had a swimming pool and there was Wofe in it, you would see the sunlight, you would see ultraviolet radiation degrading that. And over time, you would see a gradual decrease or change in the types of, science word here, polycyclic aromatic hydrocarbons. So they do break down. Hence, this is really basic, but when you get your oil changed, obviously heat and stuff of your engine is breaking down that oil. Heat, wave action, light even breaks down polyaromatic hydrocarbons in the natural environment. Not all of them.
Great. Helen's got a question about what was the total time it took you to complete this project from start to finish?
Well, I had 40 days of exposure and 40 days of spawning, so exactly 80 days. No, I'm kidding. This project was a long time coming. Obviously, when you do science like this, you don't just get out there and do it. I had to recruit the excellent talent that helped me with this project. People like Adam Kuhl or Charlie Brown that helped with this. I had to recruit people. I had to develop protocols. I had to get approval to work with animals, so that we actually, our university ensures that we're doing the right things in terms of animal welfare. And so past that, there's a lot of preparatory steps. Once you do the experiment, then there's the back end with analyzing estrogen in the blood and testosterone in the blood. I wish it was like the CSI drama where they just like pull out a vial. Oh, we just figured this out. Let's solve a case. I'd love for that to happen and be like, just wake up and be like, oh, the testosterone in this killifish is this. Unfortunately, the killifish testosterone project probably took a whole month, and then I condensed it to one slide with goofy pictures of masculine man fish. So it took a while.
Which is awesome. We'll take one last question, and this one is a really cool question from Jonathan. And he's asking, how would a college sophomore in Arkansas like me get into this field?
College sophomore, I will tell you that if I was college sophomore in Arkansas, I'd go to my university. I'd be like, let me just throw out some cool Arkansas places. Am I in Pine Bluff? Am I in Russellville? Am I in Jonesboro? Am I in Fayetteville? Monticello? Those are all academic institutions that I'm familiar with. I'd look at their biology departments. I would look at who's doing science in their neck of the woods, and I would email those people and say, hi, my name is Jonathan, and I want to clean dishes in your lab. Because that's how you get your foot in the door, and you can volunteer. Like when I was an undergraduate at the University of Oklahoma, I wanted to work with insects and insect ecology, and I just went around my building and said, hi, I'm Chris, and I want to work on bugs. And I found a guy that the guy's like, oh, well, I need some glassware cleaned. And I just started doing that for a few months, and it got me in the door to do a lot of other neat things across our country with ants and beetles. So go to the biology department, Google names, email them to see if they need help, or, not even hopefully get paid. But yeah, just write up a little resume, introduce yourself, and say, I want to do cool stuff and I want to help you do your science.
Yeah, absolutely. We've all kind of been that weird kid walking into somebody's office saying, hi.
Yeah. And, there's a few different locations that I'm familiar with in Arkansas, and you should be able to get my contact information from the LumCon website. If Jonathan or somebody else wants to email me after this talk, I know people at Russellville, Arkansas State. I used to know people in Conway as well, but I know some people within Arkansas that I'd be like, oh yeah, this person or that person, maybe, who knows?
Yeah, absolutely. So any students or teachers or other scientists out there in the audience, if you need to kind of contact Chris for his network of contacts to help facilitate connections for yourself, you can certainly respond. You have my email in multiple locations now. Just send me an email and I can definitely get you in touch with Chris. No problem.
Yeah, that'd be great. That'd be awesome. So.
Yeah.
Cool.
Chris,
anything else, Mert? This was really fun.
No, I just, we're getting a lot of thank yous. And yeah, awesome talk, Chris. Thank you so much for giving us your time tonight.
Cool.
It looks like it's getting about dark time.
Cool. And my lights are off. And also, so here's a plug. In the future, if you're interested in LumCon summer programs, I've taught fish biology there as a summer program, and it's a really awesome experience. And hopefully when we get back up in terms of say next summer and further on down the road, I hope to keep doing that kind of stuff there as well. So that would be really cool. If maybe I could see some of you guys with my fish biology class or the stuff that they do down there. Yeah,
absolutely, Chris. Again, cool. Thank you so much for both teaching at LumCon in the summers when we're able and for tonight. Just a reminder for the audience, remember, we're only 2 talks away from some of you earning those challenge coins. So remember, if you attend any 10 LumCon science talks, you will earn a research vessel Pelican challenge coin. So we're well on our way. Again, if you have any questions or want to provide feedback, you have my email and you're welcome to use that anytime. With that, my greatest appreciation for your attendance tonight, and I hope to see you again next week for Dr. Bacchus's talk about aquaculture in the United States. We'll see you next week.